Cream cheese is a popular fresh cheese made from milk or cream. Have you ever wondered what chemical and microbiological changes occur during its processing? Well, today we'll explore cream cheese fermentation, so let's get into it. For this process, the milk will undergo acid coagulation with rennet. Today we'll be using whole milk. The milk is initially pasteurized to 62 degrees, ensuring that the milk is safe and reliable for the fermentation. The milk is then cooled to the fermentation temperature. In the case of cream cheese, it is 22 to 24 degrees Celsius. The starter organisms used are mesophilic starters. Their main purpose is fermenting the milk sugar lactose into lactic acid. This results in a decrease in pH, increasing the shelf life and safety of the cheese while providing a sharp, fresh flavor to the curd. Now just stir it in lightly, and the next step is rennet. Rennet contains proteolytic enzymes, rennin or chymosin, that cleaves kappa casein. The stability of the casein colloidal suspension is weakened while releasing calcium from the casein micelles, increasing chymosin action. The loss of the macropeptide contributes to the development of cross-links between the micelles to form a moisture and fat globule trapping network. The mixture is covered and allowed to sit at room temperature for 16 hours until a curd is formed. There are two different pathways for the uptake and metabolism of lactose in lab: glycolytic or pentose phosphoketolase pathway. Lactococcus bacterium are homofermentative and ferment glucose. The glucose is metabolized to form mainly lactic acid through the glycolytic pathway. Galactose enters after the conversion of glucose to glucose 6-phosphate. Lactococci phosphorylates lactose as it is transported into the cell by phosphophenoPyruvate-dependent phosphotransferase system. Phosphobeta-galactosidase hydrolyzes the lactose phosphate to glucose, which enters the glycolytic pathway, and galactose 6-phosphate is eventually converted through the tagatose 6-phosphate pathway to pyruvate, which then produces two lactic molecules. This accumulation of acid decreases the pH of milk and causes the casein to coagulate and curdle into curds and whey. At the end of fermentation, the curd is separated from the whey using a cheesecloth, hanging overnight at cool temperatures, ensuring that the whey is fully drained from the curd. Let us now just observe the transfer of the cream cheese from the cheesecloth into a bowl so that we can fully visualize what our final product looks like. And there we have it, our final cream cheese a la Trishia. Thank you for tuning in, and hopefully you successfully understood what happens behind the scenes in cheese production.